Okay, morning all. Welcome to our SAEON Seminar. Yeah, it's the second last one for the year, so the year's nearly done, which is, yeah, it's gone by quite quickly. I just wanted to say that we're going to have Marc Pienaar talking today. He's got a PhD in electrical engineering, and he joined SAEON in 2019 as a data scientist with uLwazi. And before then, he was a senior scientist at the CSIR. And his work here at SAEON focuses on developing data pipelines through various processing algorithms to building analytical systems, visualizing and decision support interfaces. So he's been involved in quite a few projects around SAEON. And he's going to be chatting to us about one of the projects that he's working on right now on the global assessment of the mobilization of freshwater data via GBIF. So I apologize if I mute you guys. So yeah, if just keep it muted, and then there's also, we'll have time for questions at the end. And if you do have anything, you can also just write it in the chat. So yeah, I'm excited to hear more about your work, Marc.
Hi, good morning, everybody. I'm just going to share my presentation. Okay, can everybody see the presentation?
Yeah,
and
you're all good.
Good morning, everybody. My name is Marc Pienaar. I am in the uLwazi node here at SAEON, and today I'll be talking about a global assessment of mobilization of freshwater biodiversity data via GBIF. It's part of a freshwater biodiversity landscape project, which I joined in about mid-September. It's got a really strong team involved with it of freshwater specialists. I myself am not a taxonomist, so my role to date has been to investigate how best to extract data from GBIF itself, to look at patterns and trends and the various attributes that we can use. And with that, okay, so I've divided this talk into two main parts. The first is an overview of GBIF, and the second is figuring out an approach of how we could extract data using freshwater checklists that we have, and specifically using two case studies for which we have specialists in the group, namely reptiles and odonata. Okay, so what is GBIF? GBIF is a Global Biodiversity Information Facility. It was founded in 2001. It comprises participating countries and organizations that work through a collection of participating nodes. It's at the forefront of modern informatics. It has over 2 billion records. A large part of the records are citizen science based nowadays, and that's what this graphic in the top right is. It's an estimate of how many data sources are mobilizing data to GBIF from citizen science sources versus traditional sources like museums and so on, where the green are citizen science. And by far, the birding collections have more than doubled the information in GBIF. Okay, the map in the bottom right is just the map of the participating countries. Okay, so to understand GBIF, I've put it together in a sequential way from data submission up to data use and what happens in the background in between those two stages. So if you're a data provider, what you typically do is you'd organize your data and then go and download a template somewhere from GBIF, log on to its integrated publishing toolkit, and fill in the required metadata according to some standard, including compulsory Darwin Core fields. Once you've done that, you then map it to a data class in GBIF. There are 4 classes currently, ranging in complexity from metadata only, checklists, occurrence data sets, and eventually to more complex sampling event data. I understand that there's a new data model on the way according to their website, and that'll probably come into force next year. So once you've done all of this and you press register and submit using this toolkit, it goes to the background in GBIF. And as I understand it, what happens is the IPT, the Integrated Publishing Toolkit, alerts the registry via GBIF's own API that there's a new data set that's been submitted with a minimum set of metadata. That then links to a DOI of data, which creates a new DOI or uses an existing one if an agreement is already in place. And once that's done, then the crawling infrastructure, whatever that might be, kicks in. The way I understand the crawling infrastructure is it's a set of algorithms and API calls which fragment the data into smaller components and send it all over the show to its various services to do checks and balances. The most important, of course, is to match up the taxonomy that was submitted using the backbone in GBIF. And once that's all done, it gets stored in the various SQL data tables and is available for the public to use. Okay, so what is the backbone? The backbone is actually a data set in GBIF, and it's probably the most important data set. In fact, GBIF recognizes it as the most important data set. It comprises other data sets, which are ranked in order of priority, starting with the catalog of life and going on from there. The way it matches this taxonomy is algorithmic, of course. It starts with the higher order rankings, the kingdom level, and then goes through the various data sets in order of priority, starting with the checklist of life, the catalog of life rather. If it can't find a match there, it goes on to the next one and so on, until it eventually matches something. Now, here's where it becomes quite important. If it matches something at the species level as an accepted name, then it will get various keys that it uses in its relational database. And these are species key, genus key, family key, and so on. If it matches at the species key, then a universal key called the taxon key is given that species key as well, which you can use as a search. If, however, the information that you submitted was a synonym, for example, then it'll give a species key, but the taxon key, which is the universal search, will be associated with the synonym itself. If it can't match at that level and at some higher order ranking, then it'll, for instance, if somebody submits data with the scientific name odonator, so it's only on the class level, it won't get those lower order rankings, but only get a order key, and the taxon key and the order key will be the same, and it'll populate the relevant SQL databases. Okay, so here's just an overview of the backbone. This is metrics taken from the website itself. There's currently close to 4 million accepted names, about 3 million are synonyms. Most of them overlap with the catalog of life. The largest collection of information is in the Animalia kingdom, and that's mainly due to birds and the inclusion of citizen science in the last few years in GBIF. And there's quite a good match at the species level and a fair big percentage of unranked things. This might be spelling mistakes. It might be new species that don't exist in the backbone yet. Don't really know at this stage. Okay, so now on to data use. The underlying mechanism to get data out of GBIF is its API. So it's a RESTful API and it's JSON-based. What this basically means is it transfers information from its servers to some other system, in this case, the web, and the output is in JSON format. So JSON stands for JavaScript Object Notation, and this, beg your pardon, this picture in the top right is a typical output. It's a list which we can describe as a dictionary, where you've got a key and a value. The key is always static and unique, and the value is the variable, and it's also nested. So you might have a typical key such as result, which might be an array of results. These would be the records, and in those records you'd have keys and results and so on. Okay, the typical API is a URL, then it's got the API itself, the functions of that API, and the parameters you can pass to that function. So in the case of GBIF, it's HTTP API GBIF/version1. The 4 APIs it has are the registry, the species, the occurrence, and the maps. I won't really talk about the registry. It's more about creating data, in other words, pushing data to GBIF, but there are some get methods. Instead, I'll talk about the species and occurrence. But the big question people often ask is, why don't we just use the web interface? Well, it turns out the web interface also uses the API, and there's a lot more freedom when you look at a code base. In addition to that, the freshwater species we're looking at are in the thousands, and the number of records with these are in the millions. And we don't have enough time to download individual species. And then some of them are millions and millions of lines, which we have to bring into a code base as well. So we're trying to find faster ways to look through a global list of freshwater species. And that's really what this talks about, using the API to try and figure out what is the cost of searching through the API versus downloading And I should also mention that you can do simultaneous downloads through the API anyway. So it's a combination of the two, really. Okay, so in terms of the species API, this is a snapshot of some of the functions that you get in the table on the left. Here, 3 are mentioned: match, search, and suggest. And on the right, there are 3 queries. The first on the top right is search the species API. It's got the base URL, the species API, the function is match, and the name we're looking for is odonata. And when we put that in a web browser, we get a typical response, which is our name usage page. It matched odonata at the order rank, and therefore the order key and the usage key, in this case the taxon key, are the same. If we go to a lower level, at the species level, here this dragonfly, I believe it is, in the odonata order, and we get a result. It matches at the species rank, and the species key and the usage key are the same, and it had assigned the various other keys that we can use in our relational search. 2 other things to note is that the confidence of the match is 99. For this thing, this is a text-based confidence, so it's not a taxonomic confidence. It's based on an elastic search function in the background. And to illustrate what that does, the last example at the bottom is exactly the same search, but I've made a spelling mistake. So the confidence is slightly lower. It says that the match type is fuzzy, but it still managed to match it to a species. So this is just something to note, especially when you're looking at other taxonomic levels, because of the very nature and the Latin nature of taxonomy. What the web would do, it would use the same API to suggest names when you put in a search. And you just need a bit of discretion when you're looking at these things. Okay, in terms of the occurrence API, it's the same as the species API. There's just a few more functions. Yeah, I'm just using a typical one, this HTL, this URL in the middle of the page. It's the main function called search, and we pass a few parameters. Species key is the dragonfly key from the previous page. I only want to look at one record, limit equals 1 and offset equals 0. So these 2 parameters, here's the important part. The API is limited to 1,000 records and only 300 per page. So if you want to get all 1,000, you would get 300 and then change the offset to the next 300, and so on, and then aggregate these. The reason why it's 100,000 is for practical reasons, because thousands of people are using this API all the time to develop their websites, to do their science, to create plugins for various things like QGIS, for example. Anything bigger than that, you would have to request a download, but luckily there are smart ways to get around this, simply by disaggregating the information into smaller units, whether it's by year or by country or by some other unit, and then building it up again in your code base. The advantage of getting these instantaneous searches is you can explore ways to download data or extract data, but you can also speed up the amount of data you can get, as opposed to waiting for thousands and thousands of downloads, which you might have to bring into your coding base anyway. Okay, now there are clients for this. So a client is just a bunch of functions in a scripting language which wrap that HTTP request into a code base, rather. The 3 official ones that GBIF produce are Python one, PyGBIF. There's an R one, which most ecologists would use because of the additional ecology packages in R, and there's a Ruby one. But it's possible to create whatever one you want. I even experimented with a small Java one the other day, so it's very easy. But it makes life a little bit easier. So this top left here is the species API, and they've wrapped it in a function called backbone. There are a few others, of course. And to return a result, it's quite simple. So here we enter the name of a otter, and we print the results. It's a similar result to that we saw yesterday. It's matched at the species level. It's an accepted name. It's not a synonym. If we type in a synonym of that particular otter, then 2 things happen. The one is our usage key is now the synonym usage key, but our species key remains the same as the accepted name. And there's an additional field here called accepted usage key, which matches the species key. So if we combine the species API and the occurrence API, Here I've just illustrated in sequential code, where we say search the accepted name, search the synonym, and then return just the base statistics. I've made the limit 0, so we're not interested in the results. We just want to see how many occurrence counts are in GBIF. For the accepted name, there's 711. This includes the synonym. For the synonym, there's only 37. So just things to bear in the back of your mind. Now, there's two other array fields here, key definitions. One's results. This would be your 711 results, for example. And the other is facets. So what is a facet? Well, if you think about the website passes all these queries as filters using drop-down lists and text fields that you can input. And if you think about a field that might exist in the Darwin Core attributes year, for example, you would typically filter on a year. You'd say year equals 2020, and you get the results for the year of 2020. A facet, however, would look at that particular attribute field and return all values that have a result filled in. So if you say the facet equals year, all records that have a year field, regardless of what it is, would be returned. So using that, let's look at this next example. We can now say, get the otter, and then search on its family key. So in other words, we go two ranks higher, but return a facet of all fields that have a species key. Okay, and then if we sum that up, what we get, sorry, I just need to keep moving this bar around, is that there are 238 species in the family that belong to this otter, and there's a million occurrence counts that exist somewhere in GBIF. So now, by using a combination of facets and filters and logic and loops in code, it's possible to do quite a few interesting things really fast. I beg your pardon. So this little example, what we did was we created a 1-by-1 degree array. I should just note that we do have geospatialist specialists in the project, and we're still looking into the appropriate way to view these things. But for the sake of this talk, we can create this array of 64,800 individual polygons, and you can pass simple polygons through to GBIF in its occurrence search. Obviously not complex ones, because it's an expensive geoprocessing operation to do these unions, and iterate through that list using some parallel code to return the occurrence counts. So here's a map of all occurrences that have geospatial coordinates and could be returned by a little bounding polygon, about 2 billion of them. And we immediately see some trends that the majority of information mobilized in GBIF is in Europe, America, and a little bit in Australia, South Africa, and edges of South America, and so on. Okay, and then you can do the same thing for species. We can iterate through those little blocks and say, return the facet species and count unique species keys, which gives us an indication of how many species are recorded with coordinates, about 1.2 million in GBIF currently, and the same sort of pattern. We can also do other facets. So we can look at, for example, if we iterate over a year array, in this case, just starting at 1800 and ending in 2023, and we can return how many unique species have been recorded over those years, and then how many occurrences have been recorded. There's very clear, distinct pattern of an exponential increase in occurrence counts, whereas the sort of linear increase in the amount of species follows a more, I would assume, scientific approach of collecting these things. And this exponential occurrence is most likely the rise of citizen science mobilization in GBIF. We can also look at other facets. There are about 68 facets that exist in GBIF currently of the 260 attribute fields. And one of them might be number of institutes. So here, institutes are things like Sandby would be one, which submits data. Another one might be iNaturalist or the University of Bergen. And there's been quite a almost exponential increase globally of these institutes that are submitting data either actively or passively to GBIF, in the order of about 8,000 currently. And then obviously the number of countries that are collecting data sort of plateaued out at the country list in the mid-century 1900s. Okay, so on to freshwater. The freshwater species that we're looking at, or at least the checklists, come predominantly from the FADA project. That's the freshwater diversity assessment project. We have a few of those lists. There are a few old, and some of them are old. Team members are busy generating other lists. We're looking specifically at freshwater reptiles and odonata for our case studies because we have specialists of those in our team. And then some of the lists are giving problems and we don't really know what to do, like birds, for example. Okay, so exploring some approaches for this. The very first thing we thought to do at some point was use a geographic bounding box. So for things like fishes, for instance, you can pass these simple polygons, that's this map image on the right, and then return all occurrences or species or attributes that you're after, and then locally filter them through a more detailed polygon like a country and so on. That turned out to be a problem when we got to bigger countries like America, where all of a sudden we're returning millions and millions of results and it kicked us out of the API. Apologies, I just want to close the door because it's raining very loudly here. Okay, then the next approach that we did was we said, let's look into these facets and go through higher order lists. That's what the spreadsheet is, looking at various fishes and birds and other things from the FADA list. And there happens to be a facet in there, an attribute field called habitats. Now the three fields that are filled in habitat are freshwater, terrestrial, and marine. We're obviously after freshwater and iterating through all of our lists. It soon became apparent that this particular field isn't very well populated at all and not very useful, except in the instance that it might be able to add one or two additional species to our checklists, which people are otherwise doing through the literature. Okay, then going back a step, I should have had this slide earlier. Our approach so far has been to take our checklists that we've generated and push them through the species API just to make sure they exist in GBIF and where the errors are. So the additional validation step here is needed. Anything that brought out an error, we just need to investigate. Is it a spelling mistake? Is there not enough information? We need to look at these confidence matches and the synonyms and so on. So this has been done by the freshwater specialists in the team, but there are these flags to help us along. Okay, so doing the same experiment, this was our freshwater reptile list, for which we have 631 species. Obviously, duplicates are not removed in our online resource. Just for the sake of this talk, I've removed them so we can show some counting stats. Okay, using the same approach of trying to find habitats, freshwaters for all of these lists, here's the reptile example. And what I did, or what we did rather, was to say, let's go to the class level and look at reptilia and return all species in reptilia. So we get a large number of species. We see, are there any that are flagged freshwater? Out of that, only 308 were flagged freshwater, and out of that 308, only 133 were unique species. But there were 11 that didn't appear in our original reptile list. Some of them may correspond to those errors we found, but with a little bit of validation, we just need to make sure that these are additional freshwater species that we can add to our lists. Okay, then this was also just exploring some approaches. The first thing was to say, knowing that, for example, the habitat attribute field doesn't have a lot of data, let's look at all of the core. So we brute forced through a couple of these species checklists, and here's an example of one where the columns are the various keys associated with the species. And we saw, and we said, let's count how much information is filled in across all of the various tags. I think we returned something like 267 attribute fields. Of that, about 50 had reasonable data filled in, and of that 50, about 30 were about 90%. And of that 30, almost half of it was taxon related, these various keys, but there were a few useful ones, such as year and coordinates and so on. The more detailed ones, which I assume come from the more complex data sets, perhaps sampling event data sets, were very poorly filled in indeed. These would be things like depth and elevation and additional information like that. Okay, this graph on the right here, or the spreadsheet, was just another approach where we said, you know, should we try and pivot these things somehow into some or other array? This was just interesting. It was for an individual species in one of these lists where something must have happened in the 1960s, perhaps a funding project or something that kicked in place, I don't know. But this approach also turned out to be quite costly if we had to do this, you know, from the API into a spreadsheet, aggregate it, and then do this for each individual species. So a little bit costly to do in terms of timeframes. Okay, so looking at reptiles in general, if we do a global map using this approach of going through our 1 by 1 degree grids, once again, very similar patterns to what we found so far in GBIF, Europe and America and a bit of Australia and so on. And what we're showing here is all the accounts for reptiles. These are that have coordinates and are present, and there's a few other filters involved in the background, and all the species. So we've reduced it from 630 to 581. And this is due to filters that were applied in this algorithm. And then if we just look at the global trends, a massive sharp increase in occurrence counts, presumably the rise of citizen science, and the sort of linear increase in the number of species per year recorded. Okay, then on to Odonata. Here, we don't have to generate a species list individually because if I understand correctly, Odonata is pretty much freshwater anyway. So you can download the whole thing and then extract the information and species from that. This also turned out to be quite costly because a simple download is already 9 to 10 million rows. So you can't bring it in Excel, you've got to bring it line by line with a lot of redundant fields as well. So, you know, this was part of our early investigations. It turned out we could extract the same information a lot faster just using fancy filters and facets through the API. We did some initial stats, which just proved that, yes, that we can do things at the species level. We've done some other aggregate stats to say, you know, what other levels have reasonable amount of data that we're not skewing our assessment too much. And here, these two maps are just using the country level records of what's going on. So the species that exist in GBIF, according to Odonata, this is regardless of coordinates because there's a country tag at this stage. So there's a reasonable amount of species in America, South America, and some in China. But when we look at the occurrence counts, everything seems to be happening here in Europe and a little bit in America. Okay. So again, just these global trends, these facet trends that we can look at over time. There's again, this sharp increase in occurrences recorded or mobilized into GBIF. And it seems to mimic this rise of institutes that are publishing data. And my guess, once again, is that this is citizen science that's playing a big role here. So to further delve down into that, this is just a more detailed disaggregated map of the previous two. The top one is a linear scale. This is occurrence counts. And the bottom one is just a log scale, so we can get an idea of where the stuff actually is. On the top right, this is a heat map looking at publishing country and how many countries a publishing country publishes occurrence data for. So what that means is if you think of something like iNaturalist, for example, iNaturalist is registered in America. So anything that flows through iNaturalist will get the tag publishing country, but the country for which the data comes from will be the country in which it was recorded. So there's definitely a collection of publishing countries that exist here in GBIF that are publishing for a lot of other countries. And if you look at the stacked bar chart in the bottom right, the two big contributors are the Netherlands and America. And it just so happens that the Netherlands and America have two of the biggest social citizen science platforms that exist in the, in the database. Obviously, more research is needed about this. So what we have now is we have a basic approach in the background to filter through all of the various species we have. And it's about time we start doing these stats across species to look for some trends within GBIF, within species to look for unique trends and patterns, and then to start isolating countries where data should be and find out what sources are contributing to that. So the next thing that we will attempt, and we only really have this for Odonata at this stage, is a global checklist produced by a paper from Sandal et al. In 2022, where they went through the literature and the various online portals and mapped where the species should be according to these sources. That's this map here in the top left. If we compare that to the map from GBIF, there's definitely species missing in South America and China. There are some anomalies. For example, America has more species in GBIF than it does in the Sandal paper. Could be an artifact of this map being produced by a GBIF 2 or 3 days ago, and the Sandal one maybe a year or two ago. And the bottom two are just an indication using our disaggregated method to show where these things are. So what's very clear is that South America and China, in the case of, well, possibly even Congo, I don't know, we need to speak to the specialists, aren't mobilizing data as much as they should here. And our very next steps will probably be to go into these countries and extract as much information from them as possible to see why they aren't. Is the impediment financial, or do they just not have access to these various things? Is it language-based? We don't know yet. It's the next part of the investigation. So while all of this has been happening, in the background, the team's actually been working on another paper, which I see I've slipped, forgot to put its name here, which is a giant survey paper being sent out to the freshwater community in the world. We've gone through a few iterations of these surveys. I'm not as involved in this as I am in the GBIF part of the paper, but it's been translated into multiple languages. It's gone through a couple of checks, and it's about to be sent out to various experts across the world to try and find out where the issues in mobilization of data are. And together with these results and what we find from GBIF, we hope to have a nice picture of recommendations by the end of December, maybe January at this stage. Thank you very much.
Cool. Thank you so much, Marc. That was really interesting. And it's cool to see how your citizen sciences and stuff become very important in bringing the big picture of understanding where things are. Does anybody have any questions for Marc? You're welcome to unmute or raise your hand or even use the chat. Any questions?
I would prefer no questions.
There's one from Zach over here. Zach, would you like to ask your question or should I? You're welcome to unmute.
No, I just I noticed that Marc said the number of different wrappers for the GBIF API, and he didn't mention JavaScript, which made me very upset. That's all.
I'm sure there is a JavaScript one, actually, just not provided by GBIF themselves.
Cool. Does anybody else have anything they'd like to ask?
Yes, Paul Skelton speaking.
Yeah, go for it.
Thanks, Marc. I enjoyed it. I'm an old guy. I don't really dig into all this data in the way, and you obviously got a really good handle. My question is the team going beyond just those two examples that you gave? Freshwater fishes is my particular interest, and have you got those experts already engaged? Willem Kutzer, for example, and so forth. So really just where the plans in terms of diversity. Thank you.
I'll attempt to answer that, and I'm hoping there's somebody from the project team on this talk who might want to add something to that. But you know, this is an international team at the moment. It's a smallish team and a strong team, and there's two very strong experts on odonator and reptiles. But as we speak, and you know, the team's been growing and growing and growing all the time. So it's very possible that there might be an additional case study, but certainly the global trends. So we haven't gone down into the deep dives of the case studies yet. We've just been figuring out an approach that we can apply across the various freshwater species. But I can't say for certain, not having expertise in these things, which new member might be able to provide insights into another case study. And if there's somebody else from the team who has insights on that, they're welcome to add something. Otherwise, that would be the limit of my comment for that answer.
Thanks, Marc. I can jump in here. It's Michelle Walters speaking. I'm also part of this team. We are definitely going to be looking at freshwater fishes, Paul. We do have Jonathan Reddy on the team. I don't know if you've worked with him or know him. And at the moment, we're playing things by ear, seeing what we can achieve with the team we've got. And if we find major gaps, we will certainly add more people. We'll keep you in mind. Thank you, Paul.
Oh, thank you. That's quite a nice little chat. Ryan, I see you have your hand up.
Hi, thanks, Marc, and hi, Michelle. My question was linked to the BII. I don't know if you guys know that, the Biodiversity Intactness Index, which links species abundance to land cover change pressures. So the idea that as land cover pressure increases, species abundance decreases. So there's a global BII, and this was developed by Unsi Biggs and Bob Scholes, and published a couple of papers. So I was just wondering, could this approach, drawing on citizen science, help to verify some of those pressure indicators? I mean, maybe don't have to answer the question now, but it might be something worth thinking about. If you know where your pristine wetland areas are and your impacted wetland areas are, maybe you can start seeing difference in, maybe not abundance, but species richness, and it can be associated with BII type indicators. So yeah, pretty cool, and good luck for the rest of the project.
Okay, thanks. I can't answer that. I'm not an expert here, but I see there are other hands up who might be able to add to what you say.
Yeah, you can go.
Thank you very much. Ryan, yes, thank you. We are aware, and I'm wondering if you're aware that the last year's Oppenheimer grant went to someone who was developing a more fine-scale biodiversity intactness index for Africa specifically. To come back to abundance and composition, I think what we're struggling with at the moment is GBIF started as an occurrence database, and they've only recently provided functionality to include sampling events, which means repeat measures, which means you can draw on abundance and compositional changes and things like that. So there is scope for that. And in terms of citizen science, absolutely possible to launch, if you want to target a specific site, launch a little project and get the word out there and get boots on the ground for people to go see what's there, and you can repeat that over time. So you can start your own project and then please put it in GBIF. Thanks.
Cool, thanks. Yeah, I'm part of that on the peripheral edges with Haley, who's running that project. So that's why I was thinking maybe there might be some crossover, or because they're developing new sort of pressure indicators for different using experts. But now there's another level of citizen science to contribute, where there are countries that are lacking experts or areas that are lacking experts. So I just thought there might be some overlap. But thanks, Michelle.
Thanks, Ryan. That's a really neat idea.
Yeah, thanks, Michelle and Ryan. Tony, I see you have your hand up.
Yeah, thanks. Can you hear me?
Yeah.
Yeah, so my question was already partly answered by Michelle, but it was about detecting change over time and how you could use GBIF for that. And I was wondering specifically about the museum records and how time is recorded there. Is it the year that the record was entered into the database, or do they also have an attribute for the year it was collected?
Actually, it's the year it was collected, and as it's mobilized to GBIF, those GBIF only started in 2001, and if you look at those trends, there's data from the 1600s onwards. So it's the date and day it was recorded itself, and to my knowledge, there isn't actually a field on the date it was submitted. I mean, if you really go and research, you'll find when it was submitted, but it's not easily searchable. So it's more the date it was recorded itself, the event. And in terms of museum samples and citizen science and distinguishing those, it's actually a bit of a problem at the moment, unless you know which institute you're dealing with, whether it's a Smithsonian or something like that. That citizen science map I showed was actually a machine learning experiment, because they're not tagged citizen science except for the big databases where somebody, you know, translated words like volunteer and citizen into multiple languages and searched through all the fields. So it's an indication of citizen science, but we don't actually know, you know, which ones are proper museum samples unless you're working for an institute or something like that, which is another problem as well in GBIF.
Yeah, thanks for your question, Tony. Fatima, I see you have your hand up.
Hi, yes, thanks very much. I just want to share that in terms of the GBIF records, you would be able to look at the basis of record to determine if it's an observation or a specimen-based record. So based on that, you would be able to tell perhaps if it's a citizen science record, which would be specific data sets as well. So that kind of data will be available. Thank you.
Yeah, no, that's correct. Just to add that to that, there's you know, those filters, you can get out fossil specimens and things like that, but they are a little bit blurry. We did investigate some of those, and there's certainly human observations which seem to come from non-citizen science data. So it is a bit blurry still, but it is a good filter to start out with that. But there are so many of these things. It's the world of big data, and it's just getting worse. As what has been introduced lately, and I'm not a GBIF expert, but I have been going through the attribute fields, is something called I forget the tag now, but basically it's trying to distinguish duplicate records from various events. For example, if somebody did a human observation and that species died, and then somebody did DNA on it, it starts bringing these together in a you know, they're thinking about these things. And using a combination of those tags, it's possible to start differentiating out these things, but it's still a bit fuzzy at the moment, as far as I could tell, you know, trying to investigate these tags.
Cool, thank you. It sounds like it's figuring it out as you go along, basically, with some of it, and I guess they'll figure it out better eventually. Yeah, does anybody else have any other questions? Okay, cool. I just wanted to say thank you everyone for joining. Thank you so much to Marc for presenting for us, and everybody for your questions and the discussion at the end. It was really cool to hear. I just want to advertise our next talk, which will be on the 2nd of December. And it's going to be by Mfundo Bizani. He's from Alwandle Node, and he's going to be talking about zooplankton dynamics and insights from Algoa Bay, the pelagic ecosystem long-term ecologic research, PELTA. So yeah, if you want to hear a little bit about zooplankton in Algoa Bay, we'll see you next month. So yeah, thank you everybody.
Thank you, everybody.